second speaker for the afternoon is Professor Jamie Rossjohn, who is going to talk about immune sensing of vitamin B metabolites. Jamie.
So I'd like to thank the Academy for this recognition and for the opportunity to showcase some of our recent research. So my team, colleagues and I are fascinated by the immune recognition of antigens that are found on pathogens. And in adaptive immunity, there are two types of antigen recognition. That mediated by antibodies that are produced by B cells. In this particular case, the antibody recognizes the intact antigen. The focus of our research is that mediated by T cells, where T cells recognize fragments of antigens. This particular micrograph shows how the junction between T cell interacting with a virally infected cell. And it's this particular junction, or this synapse, which is encoded by a whole host of recognition events, receptor recognition events, that I'm particularly fascinated in. Breaking this down into the core components, on the surface of a T cell, you have a T cell receptor, which is a heterodimer of an alpha and a beta chain that specifically co-recognizes fragments of peptides that might be encoded by a virus that's co-presented by the MHC, the major histocompatibility complex, a paradigm that led to the Nobel Prize being awarded to Doherty and Zinkenagel. Particularly interested in understanding the structural basis of this interaction, and to do this and make use of the approach of X-ray crystallography that provides detailed atomic insights into this interaction, converting these building blocks into a pictorial representation of the interaction. This enables us to provide detailed insights into the specificity governing factors governing this particular interaction. To do so, make use of major national infrastructure, such as the Australian synchrotron. They underpin much of our research. And of course, this entire field is on the shoulder of giants, from the father and son team of the Braggs. And indeed, it's the 100th year anniversary of the field of crystallography. And during that time, many great achievements have been made. First stated, I'm particularly focused on antigen recognition, that mediated by the T cell receptor. In this particular case, you have the T cell receptor recognizing peptide as co-presented by the MHC class 1. The T cell receptor is incredibly diverse. Not only can it interact with peptide-based antigens, it can also interact with lipid-based antigens, such as that found in the cell wall of mycobacteria. And our lab has provided some insights into lipid-mediated recognition. Well, that begged the question whether there were other immune sensing mechanisms out there in human immunity. And the particular focus of this presentation is focusing on one particular axis, that of the mate T cell receptor and that of the MR1 molecule, which is an MHC class 1-like molecule. I'll go into the little detail about that in a little while. But before I do that, I'll just highlight how the T cell receptor is so diverse. So the T cell receptor is made up of multiple gene segments, such that there can be about 10 to the 8 different T cell receptors within the body. So it's composed of two chains, the alpha chain and the beta chain, and each chain is composed of multiple gene segments that are encoded from various areas along the genome. It makes the particular alpha chain or the particular beta chain. And in particular, it is these regions here that are color-coded that represent the complementarity determining regions that are the regions that interact with the antigen-presenting molecule, as depicted here. The color-coded regions are the regions that interact with the viral peptide that's being co-presented by the MHC. In addition to that, the MHC is highly polymorphic. Polymorphisms are highlighted in shaded colors here, and these polymorphisms dictate the type of peptides that can bind to a specific MHC molecule. And this is the bird's eye view of how the T cell receptor sees the peptide cargo presented by the MHC. And this is the T cell receptor's bird's eye view of how it sees a lipid that's presented by an MHC-like molecule called CD1. As you can see, it only has a small fragment which it can get a finger hold on. Now onto mate cells, mucosal associated variant T cells. They're not in the textbooks yet, and they're not, clinicians aren't fully aware of mate cells at all, which is quite surprising because mate cells can represent up to about 10% of the total T cell population in humans, and in fact up to about 45% of total T cells in your liver. They're highly abundant in the gut, hence the name mucosal associated. They're invariant because despite the diverse T cell repertoire, it has particular germline, particular T cell receptor usage depicted here. They're restricted to an MHC class 1-like molecule that's termed MR1. And these mate cells are known to play an important role in protective immunity, and they're activated by a broad spectrum of bacteria and yeast, but not by viruses. And a critical stumbling block, a critical question in the field is, what do these mate cells see? Now, most MHC molecules require a ligand for stability. We made use of this observation into how to identify the ligand. We noticed that our control, which was mixing MR1 with tissue culture media alone, sponsored the proper formation of MR1. When we analyzed the fraction of MR1 that was properly folded, we noticed it took on board a pale yellow color. So we asked what was present in the tissue culture media that sponsored this MR1 formation. We noticed that there was a whole host of vitamins, vitamin B, that are present in tissue culture media that were also yellow. So we went off to the chemist, bought a whole series of vitamin B complexes, mixed it together with MR1, and showed that we can form a fully functional MR1, as judged by biophysical profiles and also that of STS page analysis, showing it behaved very similar to standard HLA class molecules. So the question then was asked is, well, so if vitamin B, and in particular folate, what was it doing binding to MR1? Because we know folate's really important during pregnancy, as shown by the studies of Lucy Wills. So what's the Wills factor doing in terms of binding to an immune recognition molecule? So the structure of vitamin B9 was solved by Stockstad. And what we showed that when we mixed MR1 with folic acid, we didn't get folic acid bound to MR1. It was a photodegradation product, 6,4-molpiterol, shown here. The subsequent three-dimensional structure showed how the 6,4-molpiterol was closely sequestered within the antigen binding cleft of MR1. Here we show a movie showing how 6FP is sequestered within the antigen binding cleft. But 6FP wasn't stimulatory towards mate cells. So next begs the question, what are the stimulatory mate cell antigens? And perhaps 6FP provided the initial building block from which a stimulatory mate antigen could originate. And our attention was drawn to that of riboflavin. The structure was solved by Paul Carrere a while back. We also made use of the fact that supernatant of salmonella could also activate mate cells. So we purified the supernatant from these bacteria, mixed it with MR1, and to our surprise, whilst riboflavin didn't sponsor proper formation of MR1, it was indeed metabolites of riboflavin that enabled the binding of MR1. We hypothesized it was this additional factor, the ribotyl tail on riboflavin, might be the reason for the stimulatory nature of these antigens. That provided some evidence that precursors of riboflavin were mate cell antigens. So in collaboration with our chemist colleagues in the University of Queensland, we synthesized these antigens. One step back from that. Why would mate cells want to see riboflavin antigens, and what's the correlation with the possessed the riboflavin pathway, whereas the non-activating ones did not? Suggested that riboflavin metabolites the signature of T-cell activity. We could see that while 6,4-molpiterol didn't activate, salmonella supernatant did, and these particular antigens could as well. And that itself is totally sequestered within the cleft. Well, it does it by forcing the helical jaws open, a bit like an oyster revealing. What's happening is that in the recognition of microbial metabolites, such as riboflavin precursors, you get a key component of the riboflavin pathway that combines with another component non-enzymatically, such as that of the glycolysis pathway, which produces unstable pyrimidines that are trapped by MR1, which then can convert to lumazines, providing evidence that riboflavin metabolites activate mate cells. So to conclude, I'd like to highlighted that T-cell receptors can not only recognize peptides, lipids, but they also can recognize metabolites of vitamin B, and it raises the strong question of what other metabolite sensing systems operate in human-mediated immunity. So I'd like to thank the people who worked alongside me. It's been a very productive collaboration with a number of key researchers throughout Australia and internationally. I'd like to particularly draw your attention to a close working relationship I have with Jim McCluskey and his team. Thank you very much for your attention.
Thank you very much, Jamie. Questions? So, with the mate cells, Jamie, they're a you say the MR1 is a signature of bacterial infection, and they bind to the mate cells?
It's the riboflavin metabolites. So humans can't make riboflavin, but many bacteria and yeast do. So it represents a non-self discriminating mechanism.
Okay. That makes sense.
Up the back.
Jean.
This may be a naive question, but once they've seen the riboflavin, what do those T cells do? What do the mate T cells do?
They produce cytokines.
Okay, which gives inflammation or what?
Yeah, well, that's right. It's then a signature for saying there's a bacterial or there's a yeast infection. We're just on the tip of the iceberg. We're trying to understand what mate cells do in physiology and the role they play in pathophysiology. The key step was actually finding what they see in the first place.
Suzanne. Just hang on for the microphone, Suzanne.
What about commensal bacteria? Is it only things that are nasty to us that make these particular kind of T cells recognise?
No, commensal bacteria do produce them as well, but what we're not certain of at the moment is the extent of antigenic burden that's required to stimulate these mate cells and whether they also play like an immunoregulatory role.
And are there any particular human conditions that reflect perturbance of this particular system?
That's what we're actively pursuing at the moment. There's been some reports of mate cells playing a role in irritable bowel disease, but to be able to establish that, you need to be able to track mate cells ex vivo, develop tetrameric reagents to do that, and we've done that. So we're now poised to undertake a number of studies on mate cells in health and disease, but diseases of the gut are prime suspect.
Our last question, Eugenie.
A very naive question. What happens if you have a high intake of riboflavin?
Well, riboflavin itself is not recognised by mate cells. It's the precursors. What's not clear at this stage is what happens if riboflavin breaks down, whether that could potentially represent antigens. That's something we're aiming to take a look at too.
Please join me in thanking Jamie for a very interesting talk.